Okay, hi everyone. Um, so I'm still kind of fussing with my new computer setup because it's kind of confusing. I can't figure out how, when I'm setting it up, I'm supposed to get the Zoom screens to look right. So if during the lecture, if it seems like the slides are not actually progressing, just shoot me a chat message. I have my little chat box open, and I can try to troubleshoot. Okay? All right, but hopefully it'll be okay. All right, let's move on and switch gears and talk about the cell cycle and mitosis. Right? So the cell cycle, of course, are the different stages or phases of a living cell as it performs its normal everyday functions and also maybe participates in dividing, right, as part of reproducing that cell. So that actual process of dividing we call mitosis. So let's look at these two concepts together as we introduce thinking about these things. So here is an image of some cells. These are cells at the tip of a growing onion root. All right. And so at the tip of a growing onion root, as you can imagine, there are many cells that are actively dividing as the onion root grows into the soil. And so it's a nice opportunity to take a look at cells at the tip of the onion root that might be sort of captured at different moments doing different parts of the cell cycle. So many of these cells, most of them in the picture here, might look like this one here or this one here. So this would be your typical cell just doing its normal differentiated cell functions, right? Carrying out whatever tasks it's programmed to do. If you look inside the nucleus, as we've come to appreciate over the course of the semester, the nucleus looks very grainy. That's because it's filled with the unraveled chromosomes of the onion. And each chromosome of the onion represents a strand of DNA that contains potentially thousands of different genetic sequences on it, the genes, right, that are part of the onion's genome. We learned that at any given time, certain genes are being used by the cell, certain genes are not being used. And as a result, you might have regions of the chromosomes that are more loosely sort of opened up, right? The more euchromatic regions of the chromosome. So that would look like the more lightly speckled regions in here. And you'd have certain regions of certain chromosomes in this onion cell that might contain genes that are just not being used at that time, right? And those might be regions of the chromatin that are more packed up and tightly held as heterochromatin, to discourage regulation of the genes in there. And so those might be the darker regions that you see here. So as you look at these cells, you can see, right, different cells look like they are utilizing different genes of their genome. Right? So those would be cells that are doing their normal differentiated functions. But what about these other cells in here, like this one here? Right? What a neat kind of image. Right? These are the chromosomes that are condensing, right? So rather than being all loosely kind of just kind of in the nucleus, right, taking up different areas of the nucleus, these chromosomes have started to condense. So you can actually begin to really make out one chromosome from another. Look at these down here towards the bottom. These have condensed even further, right? So they're really becoming like these thick sort of chromosome rods. Look at this cell over on the upper right here. These rods have really condensed and they seem to be lining up in the middle. Some sort of like imaginary line, right? And then look at these over here. Right? Two sets of chromosomes are now separating, right? Separating so that the cell is well on its way to becoming two new onion cells. Right? And in fact, this pair of cells right above it may represent, right, a couple of daughter cells that have successfully now emerged from the process of mitosis. And what do you see? You see that the chromosomes are still pretty condensed, but they're beginning to unravel. Right? And then maybe at a certain point, they will then resume their normal differentiated state, and you'd have two fully functional new onion cells. So really cool image, because you can see these different parts of the cell cycle and mitosis represented here. So one thing to keep in mind, and we've discussed this previously, but I think it's important to think about. At any given moment, a living cell will either be busy, sort of preoccupied, carrying out its functions that are determined by the genes. And so it'll use the genes that it's meant to use from the genome that make it, for example, not just an onion cell, but specifically an onion root cell, right? Which might use different genes than the leaf part of an onion, right? So different functions during this stage of the cell's life include things like biosynthesis, making complex biomolecules, catabolism, breaking down molecules to get energy. The cell might be sending out signaling molecules or other types of cell type specific work. On the other hand, just like in the onion cell slide that we saw before, some cells are not doing their normal function in that photo. Those are focused on replicating its genome and dividing. So that has to do with reorganizing the chromatin, being able to replicate the genome that's inside the cell, so you have two copies of it, duplicating all the cell components that also need to be shared if this thing is going to split into two cells, and then the actual physical separation, right, from one parental cell into two daughter cells. Importantly, the cells cannot be focused on both sets of these tasks at the same time. Right? Why? I think on an intuitive level, I think you can understand why, right? Because the mechanisms involved in carrying out normal differentiated cell functions, for example, choosing which genes to use, getting transcription factor proteins to bind to the different regulatory parts of those genes, like promoters or enhancers, and then eventually getting RNA Pol2 to come to transcribe the genes. Right? Those complicated mechanisms are very different than what takes place when you are replicating DNA. Remember, when you replicate DNA, for example, you can imagine one single chromosome, right? You're going to have lots of places where the two strands of the DNA separate, right? Those are the replicons or the replication bubbles. And as those bubbles grow in size, right, within those bubbles, you have things like DNA polymerases coming in, right, being able to copy the original template strand to make a new copy strand. And then remember, that would be like on the leading strand. On the lagging strand, it's even more complicated. You're putting down random RNA primers in order to be able to copy, the template strand, sort of going backwards by making Okazaki fragments. So think about those two processes that we've been studying. Yeah, you can't have that happening at the same time, right? There'd be too many problems, because how do you control the regulation of genes, right? By tightening or loosening DNA and chromatin to allow transcription factors to get in, if at the same time you're just separating all the DNA. Right? That's just like your car, right? If your car needs transmission work, right, you have to bring it to the mechanic and leave it there. Right? The mechanic cannot just hitch a ride with you as you continue driving your car and do all the repairs needed on your transmission. Right? It has to be one or the other. Okay. When we think about a single cell dividing, I think a great way to think about the purpose of cells dividing is to think about simple organisms, simple single cell organisms. Right? For example, prokaryotes like this, well, not prokaryotes, but single celled protists like this amoeba. Right? So an amoeba is a single celled organism, but it is a living thing, right? And so its whole being is just contained within its one cell. So it's a eukaryote, right? So just like eukaryotic cells in our body, And so inside of the amoeba, you would imagine to have different organelles, for example, a well-defined nucleus. You would have this contractile vacuole, which serves to help manage the water inside the amoeba, so the amoeba is not always swelling up. Right? And you have little vacuoles where it's taking in food particles, and maybe now it's breaking down these food particles. And you have mitochondria, right? Mitochondria that takes care of doing aerobic respiration. So here is a microscopic image of the amoeba, and you can see some of these features inside of it. Right, here's the contractile vacuole. These brown spots might be other features like mitochondria, and lots of other interesting sort of specks in here, right, which represent all kinds of different structures and protein groups that are in there. So when an amoeba reproduces, it simply divides. It divides into two. And that's what you see on the right here. So this process of a single-celled organism dividing, right, its whole reproductive process is just binary fission. It just breaks into two or splits into two. Right? And each one of these daughter cells contains a full copy of the amoeba's genome, as well as a shared portion of things like mitochondria and all the other goodies that are needed to be in there, so that once these two daughter cells fully separate, each one represents a new amoeba. So why does something like an amoeba even divide in the first place? Right? You know, you might say, well, that's obvious, right? Because that's how it reproduces. Yeah, but it's just a single cell. It doesn't have a brain, right? It doesn't think about anything, right? So what is its sort of incentive to even divide in the first place? Well, for single-celled organisms, when we talk about growth of the organism, we're basically just talking about it's obtaining nutrients, more and more nutrients over time, right? As it's maybe in the pond water or wherever this particular amoeba lives, right? And so it is successful in being able to create large macromolecules inside of it, right? Taking simple things like nutrients, building things like carbohydrates, lipids, nucleic acids, and of course, proteins. Right? So over time, the more successful this amoeba is, it's simply going to get bigger and bigger and bigger, right? As it's kind of building up all of these wonderful macromolecules. The problem is, the bigger the amoeba gets, right, the more spaced far apart these different components are inside of it. Right? So this is kind of funny, right? You don't think of this as a practical consideration, but if a cell gets too large, right, the efficiency of all these different things taking place inside of it, right, begins to take a hit. It gets harder for things to efficiently work together. So therefore, there are limitations to how large a cell can be before it encounters these problems, where things are literally too far apart inside for things to work well. Therefore, reaching a certain size might stimulate a shift in the amoeba from the normal behaviors of kind of roaming around and eating and doing its metabolic functions, right? Switching over now to focusing on division. So I think you can sort of appreciate, right, in this case, right, that's a good example of how either the amoeba is concerned with doing its normal functions or it's concerned with dividing, right, but not both at once. Okay. So here's an interesting question. Right? You know, I sometimes like to pose these thought questions to get you guys thinking about it. If in this photo, the two resulting brand new amoebas are really just sort of shared parts from the original starting amoeba, then in a way, doesn't that starting amoeba still sort of exist, but now as two separate amoebas? Right? In other words, where did the life of that starting amoeba end, and where did the life of these two individual new amoebas begin? It's a very blurry line. And consider this also, that starting amoeba itself came from where? Right? It was one of two new amoebas that came from its own parental amoeba. So you can keep going backwards in time. So here's a question. Are amoebas immortal? Right? Now, obviously, you might say, well, no, because if some other organism comes and eats the amoeba and digests it, that's the end of it. Okay, right? Or the little puddle that the amoeba is living in dries out and then it ends up baking in the sun. Okay, that's the end of it. But all things considered, right? Are amoebas immortal if they're allowed to just keep going in their ideal sort of environmental conditions? Where does life begin? Where does life actually end? So it's one thing to have some fun thinking about this in terms of a single-celled organism like an amoeba, but what about us, right? We are these big, complex, multicellular organisms, right? What are some of the reasons why the cells in our body have to divide? So the three main reasons that the cells in our body divide are, of course, to allow our bodies to grow. Right? It allows us to replace lost or damaged cells. Every day we lose a lot of our cells just by design. Right? For example, the cells of your gastrointestinal tract, starting from the cells, the cheek cells that line the inside of your mouth, right, all the way down, cells of the esophagus, stomach, intestines. They are designed to be shed. And we do, in fact, lose many of those cells every single day. Just the physical act of eating something, right, rubbing against the inside of your mouth is going to shed some of those epithelial cells. So the fact that our body is able to replenish those lost cells, right, certainly involves cell division somehow. And of course, if we get injured, right, if our tissue is damaged, then certainly cells must divide to be able to restore, right, or heal the damage to our bodies. And the third important consideration is cells must divide, the cells in our body must divide as part of the process of passing genetic material onto our offspring. Right? And that type of cell division is a very special type of cell division. Right? That is not just mitosis, but it involves a special step of cell division known as meiosis. Right? That only the gamete cells, the reproductive cells in our body, do meiosis. Right? So we'll get to meiosis on a later lecture. So let's think about this. On the lower right-hand corner of the slide is an image of some cheek cells taken from the inside of the mouth. And these cheek cells, they're epithelial cells, excuse me. And these are an example of some healthy ones here. You can see the dark pink thing is the nucleus. Right? So these cheek cells all have the same genome in it from the person that they came from. And each cheek cell, just like every nucleated cell in the rest of our body, right, has the same copy of the genome that we inherited from our mom and dad. This raises an interesting question. So if, let's say, a cell contains an exact copy of your complete genome, the human genome that you got from mom, the human genome from your dad, every nucleated cell in our body has that. And that provides the blueprint, the master blueprint that makes you, you. So does that mean a cell from your body is uniquely you? Each of our cells is a living thing and carries with it our own unique genome. So if you think about it, what about the amoeba, right? The amoeba is its own living thing and it carries around its own unique copy of the genome. An amoeba, when it divides and becomes two cells, right, it is two complete amoebas. If each cell in our body has the genetic potential to become any other cell in the body because it contains the same copy of the genome, does that mean that each of our cells is actually us? And you might say, well, yeah, isn't that true? Because a cell from your body with its genome, right, is uniquely you compared to the other 7 billion people on this planet. But how far can you take this concept of the cell being you? For example, so let's do this, right? If we took a Q-tip and I, let's say, swabbed the inside of my cheek, all right, got several thousand epithelial cheek cells to come off, right? It might be something like this. So this would be a population of different types of skin cells from the inside of my mouth. Some of them are differentiated differently than others. In other words, the lining, the most outermost lining of cells might be more keratinized. They might express more keratin protein, right, to be a little bit tougher than the cells that are like the next layer down, which might not be as keratinized. And so that's why in this image here, they've stained for different proteins in these cheek cells that came from a Q-tip swab. And you can see some of them represent the outer layer cells. So they would be the keratinized cells, like these yellow ones here. Right? Whereas you have these other pink ones that are the non-keratinized ones. Right? So each one of those, right, would have a copy of my genome in it. If I go ahead then and dip the end of this Q-tip that has these cells on it into a flask, right, that contains really rich tissue culture media. So this is media that has all kinds of nutrients in it, like amino acids, like carbohydrates, all of the sort of raw materials that a cell would need to be able to continue surviving. Right? And let's say I take this flask and I go put it in a 37°C incubator, right, that has a proper gas flow of oxygen and carbon dioxide to simulate the conditions inside of my body's tissues. What I might find is that a few days later, if I took this flask out and looked under a microscope, I would see that these cheek cells not only are continuing to be alive, right, but maybe some of them are even dividing. Right? And I would say, oh, this little flask, right, it contains tissue from me. So is that actually me? Right? If I then say, oh, that's fun, I put it in the incubator, and then on my way home, you know, I'm driving and something really horrible happens to me, right? And let's say I get killed, right? If I get killed within a few hours, all of the cells in my body would have been deprived of the oxygen they needed. All the cells in my body would have died, right? But then what, a week later, some students remember, wait a minute, there's that flask in the incubator in the lab, and they go and they look right under the flask and they see all of these cells growing, right? So is that me in the flask? I ask that because the idea of course, of taking something like those cells in the flask and actually being able to reproduce a full human being from it by reprogramming the genomes inside of those cells to de-differentiate and become more stem cell-like, and then encourage them to become all the different types of the cell body again, cells in the body again, you know, we're still quite a ways from that, right? So right now it's still science fiction. But I think from everything that you've learned during the semester about gene regulation and about differentiation, you know that it is theoretically possible, right? To take that flask that I happened by chance to leave in the incubator, right, and actually grow a new human being from it with that unique genome that was my genome. So would the unique human being that's grown up actually be me? All right. So this again highlights the important concept that cells are either concerned with dividing or they are concerned with performing the different differentiated cell type functions that they're assigned to do, right? Utilizing the same genome as the blueprint to get all the different instructions in the form of different sets of genes that they are told that they should be using. So the difference between stem cells and differentiated cells in our body very much highlights this. For example, we already talked about how in our adult bodies, right, you and I participating in this class, right, we no longer have any pluripotent or totipotent stem cells. Right? The originalized fertilized egg, right, that represented you at the moment of conception, right, with the genomes of mom and the genomes of dad coming together, that is the ultimate totipotent cell, right? Because that one cell over 20 years of your life leading up to this point has become, right, the trillions upon trillions of cells that are you sitting there today, right? we said that in our adult bodies, we no longer have those totipotent or highly pluripotent cells. What we have in our tissues are the multipotent cells. That is, cells that have gone far enough down the sort of decision-making tree of different sets of genes turning on and turning off, right? That they then can only become a handful of fully differentiated cells from that point on. Right? That was the whole origami analogy, right? At a certain point, once you've made enough folds in your origami paper, you're sort of limiting yourself, right, to only a subset of possible eventual shapes that you can head towards if you've already gone through this number of folds, right? So multipotent stem cells, for example, the ones in our bone marrow, they are not concerned with being differentiated cells themselves. Their concern is to repopulate and provide more cells that can become differentiated cells. So their concern is just dividing. So at this point here, right, multipotent stem cells will divide, and that's all they kind of care about. And then one of the daughter cells of that stem cell sort of maintains its stem cell-like property. So that represents, right, the sort of continuation of this stem cell going on. Whereas the other daughter cell begins to divide rapidly, and as it's dividing rapidly, it is now kicking off different programmatic decisions, right? The different folds of the paper that are beginning to now give the origami shape is now beginning to give these daughter cells of each subsequent generation more and more differentiated commitments to what kind of cell it would be. So that's how this one multipotent cell, right, can give rise then to all of these numerous different types of fully differentiated cells. And importantly, these are all differentiated cells that represent blood cells, different types of blood cells, right? Because that's all that this multipotent stem cell can still change into. Right? It cannot turn into a nerve cell or a muscle cell, right? Because that time has already passed. Those decisions where it would branch off of the sort of cell differentiation tree, right, happened, it passed that already, right? So it can still go on to make blood cells, but that's what it's sort of limited to. All right. So that brings us to an overview of the cell cycle itself. I think all of you realize that this idea of a cell cycle isn't really the cell going around in a loop or doing something, right? It just represents changes within the cell that reflect that it's either doing its normal differentiated functions, or it has now shifted and it is now working on doing things like replicating the genome and eventually heading towards mitosis and dividing. So there are four main phases. For convenience sake, we can talk about these in terms of pretending like one complete cell cycle is a 24-hour day. That's absolutely not true in real life, because different cells, I'm sure you can already figure this out, different cells divide at different rates depending upon, their job in the body. For example, the cells that replenish our skin cells, those divide a lot. Whereas the cells that make neurons, or the nerve cells, those divide very little. Right? But for convenience sake, let's pretend that this cycle is taking place in 24 hours. Right? So the first phase is the G1 phase, or GAP1 phase. So most differentiated cells will potentially stay for the rest of their life in G1, just doing their normal differentiated functions. But a cell that might still divide a couple times more, right? It might still spend most of its time in G1. So in a 24-hour day, we'll just pretend that 11 hours out of that day is in G1 as the cell carries out its normal differentiated functions. If the cell is stimulated to begin dividing, right, the first thing it's going to need to do is copy all of its genetic information. So that happens during S phase, or synthesis of a brand new set of chromosomes, right? So it's DNA synthesis. So that takes about 8 hours. Right? So your cells take about 8 hours to fully copy your genome through the DNA replication processes that we've learned about. Then G2 or GAP2, during this 4-hour phase, the cell starts producing or expressing certain sets of genes that it's going to need to make protein products of, because those proteins are going to be really important for mitosis. And then the 4th phase is mitosis itself. And mitosis actually happens pretty quickly. Once mitosis starts, if everything goes well, the whole process takes about 1 hour. And we will then focus in and take a closer look at the different parts of mitosis. But first, let's look at this. So these are chromosomes in this karyotype. Remember, a karyotype is an image oftentimes used by clinicians or pathologists to try to identify any types of genetic abnormalities that might be responsible for a patient's condition that he or she is presenting with. So this is a nice image because it shows you we have 22 pairs of autosomes, right, and 1 pair of sex chromosomes. So therefore, one cell in our body has 46 individual chromosomes in it, right? If the cell is in G1, right, normally just doing its normal functions. Right? Notice though, a couple things to keep in mind, right? Each pair, right, one came from mom, one came from dad, right? You guys know that. But notice in this particular image, right, each one, the one from mom and the one from dad, right, has already replicated itself. And the copy of mom's chromosome 1 is still sticking to the original chromosome 1 that came from mom, and the copy of chromosome 1 that came from dad is still sticking to the original chromosome 1 that came from dad. And that's why not only do you see a pair of each chromosomes, one from mom and one from dad, but you see each chromosome within that, right, is actually a pair of sister chromatids at this point. So that means this image was taken when the cell was well into mitosis already, right? DNA replication had happened during S phase, and now the chromosomes are really condensing to make it much easier to be able to keep track of them. What do I mean by keeping track? One of the important reasons why chromosomes condense into these thick, stubby rods during mitosis is that it's just physically much easier to be able to now divide them up and share them between the 2 daughter cells that's going to form, right? Think about it this way. In this course, I've sort of made an analogy of thinking about your chromosomes during G1 as being like a bowl of Simon noodles, right? Because all the chromosomes are just kind of wavy. They're all sort of like all over the place, tangled together. Right? Imagine that, you and a friend, right? You're studying together and it's time for a snack break and you're like, hey, I'm going to boil up some Simon, right? And you say, you want one? You want a pack? I'll have a pack too, right? So you go to your pantry and you get 2 packs of Simon, right? And you tear each one open and you take out the square of condensed, dried, Simon, right? At that point, it's much easier to be able to say, okay, you're going to get one and I'm going to get one, right? Once you put them both into the pot and boil them all up and everything's tangled together, right? Then if you go ahead and share them into 2 bowls, imagine how nearly impossible it would be, right? To actually say, these noodles that I'm putting in your bowl, they came from this particular rectangle of Simon, whereas the noodles that I'm taking out of the bowl and putting into mine, all of those noodles came from my square of Simon, right? It'd be really hard to do that, right? So that's why if you really need to separate things cleanly, the chromosomes being divided up into 2 daughter cells, right? It's much more practical to get the chromosomes to condense first. Right? In the case of you sharing noodles with your friend, I mean, you might tease each other, hey, you got more than I did, right? And you just kind of joke about it, right? But in real life, if it's a cell that's dividing into 2 daughter cells, it's a big deal, right? If the chromosomes don't divide up evenly and you have one cell that got too many of the chromosomes and one that got too little, because guess what? That's not going to work, right? Okay. So let's look at the process of mitosis more closely here. So the 5 stages of mitosis are prophase, prometaphase, metaphase, anaphase, and telophase. Depending upon what book or what source you're using to study from, they may end up grouping prophase and prometaphase together and just call it, prophase, like early versus late. All right. But just keep that in mind. But there are some reasons why sometimes it's useful to think about prophase and prometaphase as being separate. So you can see here, during the normal part of the cell cycle where the cell is in G1, and as it's going through DNA replication and also through G2, right? All of those parts of the cell cycle we call interphase. So another way of thinking about it is that a cell is either undergoing mitosis, right, which is the M phase, or it's somewhere else in the cell cycle, which we call interphase. And so that's why down here in this cartoon, right, most of the whole cell cycle through G1, S, and G2 is called interphase, and then in yellow is mitosis. Right? So leading into mitosis, the chromosomes are condensing. Right? So the noodles are kind of condensing now back to a nice compact, these rods that make it much easier to then keep track of. During prometaphase, right, they are going to condense very tightly into these X's that you see, and they will attach to these different protein structures, which are these microtubule filaments known as the mitotic spindle. During metaphase, these spindle fibers will actually line up all the chromosomes along an imaginary central metaphase plate. During anaphase, these microtubules are then going to start shrinking away and pull the sister chromatids apart. So each one of these, let's say mom's chromosome 1, that has a copy of mom's chromosome 1 still stuck to it. That's why it was like an X-shaped, right? These fibers are going to then pull these apart so that you now have an equal division, right, of the genome. And then step 5 is telophase and cytokinesis, the actual separation of the cytoplasm and the cell membrane to form 2 new daughter cells. Okay. Let's take a closer peek. So first, let's look at interphase, prophase, and metaphase. So again, during interphase, especially in the G1 part of interphase, the chromosomes in the nucleus just look like a bowl of noodles, right? You cannot really tell where one chromosome is, where one ends, where one begins. Right? And you have a very well-formed nuclear envelope around it, defining the nucleus. During prophase, you can see what is happening is that the chromosomes have condensed now, right, to get more of that sort of rod-like shape. So keep in mind, during interphase, as you went from G1 into S phase, each one of the chromosomes, right, began condensing and began to replicate, right? So that's why each one of the chromosomes here, you can see, right, has a pair of identical chromosomes still attached together. You also see the start of forming this mitotic spindle. So up here in this image, this is a cool image, right? You can see the mitotic spindle fibers beginning to form, right? And inside the nucleus, you can see the chromosomes are really beginning to condense. Then the latter half of prophase, known as prometaphase, is you see the connection of these spindle fibers, right, to these chromosomes. And you begin to actually see that 2 sort of poles are beginning to form, right? 2 ends of the cell beginning to define where the 2 separate daughter cells are going to be. And that has to do with these special structures known as the centrosomes. So the centrosomes are made up of these little cylindrical structures known as centrioles. And they are like the anchor point that defines the 2 sides of the cell. And these are the 2 sides that are going to become the 2 separate daughter cells. Okay. So here you see the attachment of the chromosomes to the mitotic spindle. So these are the spindle fibers that would be coming from those centrioles and centrosomes on either side that are reaching towards the middle and attaching to each sister chromatid pair. So let's pretend that this is chromosome 1, and it's chromosome 1 that came from mom, right? So maybe the one on the left, let's just pretend it's the original one. The one on the right was the copy that was made, right? But they're still kept together to help keep it organized, right? So the spindle fibers will attach to special protein complexes known as the kinetic core at the center of the sister chromatid, the X-shaped structure, right? And they're going to attach there so they can then pull these two apart. For many of the cartoons, for illustration purpose, to keep things simple, right, the cartoons often just show a pair of chromosomes. In other words, rather than the human genome being represented as 22 pairs of autosomes and a pair of X chromosomes, I mean, the cartoon could show that, but it'd just be really messy and confusing, right? For argument's sake, let's just pretend that the genome just consists of two chromosomes, a big one, and a small one, right? So that's why you see here, you have a big one and a small one, right? And this big one, maybe the dark blue one represents the one that came from dad, right? So so you have this dark blue one that came from dad, this one that came from dad, and it's now replicated. And you have a large one that came from mom, a light blue one, right? So let's say this is chromosome 1, and maybe this is like chromosome, I don't know, 17, like a small chromosome, right? You can see how the fibers are connecting, and it helps line them up along the middle. So in this image here, the various proteins that make up the kinetic core are stained in a fluorescent marker that fluoresces this nice bright pink. So you can really see that each one of these spots is where spindle fibers would be attaching and arranging the highly condensed chromosomes in the middle. So then metaphase is when these chromosomes are as condensed as they can be. This is really now when they're the little thick, stubby X's, right? And they're lined up along the middle. Again, keep in mind, right? This is chromosome 1 from dad, this big one down here. This is chromosome 1 from mom, right? This is maybe chromosome 17 from dad. This is chromosome 17 from mom, right? We are just ignoring all the other chromosomes just to keep this cartoon simple. During anaphase, the spindle fibers begin to retract, right? And they pull on the two sister chromatids, therefore separating them, and they begin to pull them to either centrosome on either side, the anchor point. Right? So that's anaphase. Telophase then is the nuclear envelope begins to reform around the two new forming nuclei, right? Each one with an equally shared copy of the original genome in it. And finally, cytokinesis, right, where the cytoplasm in the cell membrane itself, right, then separates and divides. So importantly, mitosis is all about making sure that the two resulting daughter cells have the exact same genome as the original starting cell. Right? Because when your skin cells are busily replenishing skin cells, or your hematopoietic stem cells in your bone marrow, right, are dividing, right, to repopulate more differentiated cells, you're not actually messing with the actual contents of your genome at all, right? Your cells want to make sure that every new cell that comes out has the exact accurate copy of the genome that it's meant to have. Right? Okay. All right, so this is the last slide, and we'll end here today. This is kind of an antiquated, interesting sort of thought experiment here. There is classically in developmental biology this very antiquated, old school sort of saying of ontogeny recapitulating phylogeny. What does that mean? Ontogeny refers to when a cell, like an egg cell, is fertilized by sperm and an embryo begins to develop. Ontogeny, right, means the different types of shapes of the developing embryo as it grows and develops and eventually becomes a new organism that's ready to be, let's say, born. Right? Phylogeny, of course, represents a different kind of history, right? Phylogeny has to do with evolutionary history, all the different types of organisms, animals, creatures, whatever, that came before, that eventually led through eons of evolution to reaching this particular animal that you have today, right? Whether that's a fish, a reptile, a bird, or a human, right? Because I think you're probably aware that if you look at a human embryo early enough, right, in gestation, right, as it's developing, it still maintains many of the same morphological features as things like birds, reptiles, and fish. For example, we still have tails, right, that don't look much different than the tail of a bird embryo or a reptile embryo, right? We still have gill slits, right, that look very much like the gill slits of fish. So that's why, in the 1800s, as early biologists began to look at embryonic development, right, there was this kind of romantic notion that in reality, as we develop into people from a fertilized egg, that we somehow visit, right, along the way, becoming the different types of animals that are in our evolutionary past, like fishes and birds, right? Now, of course, that's not literally true. Right? I think you know that. We don't actually go through phases of being a reptile or a bird as we develop into a human fetus, right? But there's no doubt that our genomes do carry echoes of our evolutionary past all the way back to the earliest single-celled eukaryotes. Right? For example, let's end this lecture just thinking about, once again, our friend, our ancient ancestral cousin, the amoeba. Right? The amoeba represents one of the earliest forms of eukaryotic cellular life on this planet. Right? And so it's very much true, right, that over eons of evolution, right, that is what eventually gave rise to multicellular organisms, mammals, and eventually us. Right? So here you have a couple of amoeba, right, that we noticed here. Right? Yeah, right, there's these sort of blobs, they kind of wander around, right, they change their shape, they have things like a nucleus, different types of mitochondria, vacuoles in it, and they're just going around eating things, right? They're eating things by kind of wrapping their cell membrane around them and consuming them, right? But you know what? Not so fast, because I kind of lied, right? Because these are not actually two pictures of amoeba. The one on the left is an amoeba, the same type of amoeba that we looked at earlier in the lecture. The cell on the right is a macrophage, right? A human white blood cell. Now you have macrophages. They're a very important part of your immune system. They crawl around inside of your tissues looking for things like bacteria, viruses, or even bits and pieces of old cells that are broken down to consume. How do they consume them? They wrap their cell membrane around them and take things in, right? And how do they wander around? They wander around in the same way that amoebas do. As it turns out, the genes that make a human macrophage, right, that are all part of our of the genome in our cells in our body, right? These are human versions of those same genes that amoebas have. So through eons of evolution, mutations as part of natural selection over hundreds of millions of years, right, have changed these genes so that they are now human versions of them, but they are still the same genes that the lowly amoeba had way back then. So all life on Earth is connected by this thread of cellular evolution. And that's the thought that I would like to leave you with today. And next time we will continue our discussion about cell functions. And so we'll do that on Friday. Okay. All right. Let's go ahead and stop there.